So our next speaker is Duarte Barral. Duarte has a PhD in cell biology from Imperial College London. She was a postdoc at Harvard Medical School, and he is currently a Turner Associated Professor with habilitation at the NOVA Medical School. He has been working in the field of membrane traffic. His group has also been studying the molecular mechanisms of melanin secretion, transfer, and processing, as well as the role of lysosomes and lysosomes exocytosis in cancer progression. So, Duarte, welcome to our virtual seminars, and thanks a lot for accepting this invitation.
Thank you very much for the invitation. It's a pleasure to be here. So I'll share my screen and here. Okay, so today I'm going to talk about these 3D models of skin and epidermis for studies of pigmentation, diseases, actually disease modeling, and cutaneous melanoma research. So, as you are all aware, skin pigmentation is a feature that distinguishes us. Classically, there are six. The classification of skin pigmentation has 6 phototypes, and there are also some diseases like albinism, vitiligo, and others, like rare syndromes, for example, this one that I'm showing here, that I'm going to talk about later, Griscelli syndrome, that affect pigmentation, and they are highly impactful. So we've been studying for many years now what are the molecular and cellular mechanisms of skin pigmentation. We know there are 2 main cell types involved in this function. Melanocytes, these cells here that are dendritic, they have dendrites, they produce melanin within melanosomes, and they transfer the melanin to adjacent keratinocytes. So the epidermis is divided into several layers or strata, normally 4, that are represented here. Then we have the dermis, on top of which the epidermis is settled. And here you can see a histology of the human skin with melanocytes full of melanin here at the base, at the basal layer, and also the keratinocytes. And you should see that keratinocytes have these caps of melanin, these supernuclear caps, which are also very interesting, and we are also studying them. So we are focusing mainly on the crosstalk between the 2 cell types, melanocytes and keratinocytes, and how this crosstalk is important for the skin pigmentation, and especially in the process of the transfer of melanin from a melanocyte here on the left, then right, to a keratinocyte here on the right, and you see here the melanin granules. So this is known for a long time. You see here, for example, a paper from 1964. This was already observed, but what we didn't know and only recently has been clear is the mechanism by which this melanin is transferred from melanocytes to keratinocytes. And we've studied this in the past, and we are still studying from the secretion to the internalization to the processing of melanin inside keratinocytes. And here you can just appreciate that this is an old schematic, but you can appreciate that we know much more, we still know much more of what happens inside a melanocyte here in blue than what happens inside the keratinocyte. And the transfer process has remained elusive for many years until recently. So we know a lot about this formation of melanin inside melanosomes. We know that melanosomes have several stages of maturation, and then we know that they get across from melanocytes to keratinocytes. What we didn't know was how and what happens inside the keratinocytes. But actually, for example, for this to build this schematic, there are many, of course, many studies involved on this. And for example, some of the machinery for example, this machinery here, these complexes that are involved in melanin production inside the melanocyte. Mouse models were instrumental, and you can see here a collection of mouse models that have coat color deficiencies, so they have different shades of gray compared to the wild type. The wild type is the black 6 mouse, and for many years mouse models, so the mice were essential to define the machinery that I just showed you, the machinery that is involved in melanin production. But it turns out that mice don't pigment the skin, they pigment the fur. So the melanocytes are in the hair follicles, and they produce melanin, and the fur gets pigmented, of course. But if you look, for example, at the mouse paws, you see that the skin is not pigmented, so it's the fur. And if you shave a mouse, you see that the skin is pink and is not pigmented. So they are not really good models to study skin pigmentation because they don't pigment the skin, and we are interested in skin pigmentation, not in hair or fur pigmentation. So this shows you the models of melanin transfer proposed in the literature. There are 4 models. I'm not going probably to explain in detail, but you can see here different types of transfer that were postulated along the years. And we actually used, some time ago, we used skin biopsies to check which model is predominant, at least in human skin. And for that, we used, as I said, skin biopsies. We did electron microscopy studies and also optical microscopy studies. I'm not going to show all of them because this was published some time ago. But here you can see a good example of the interface between a melanocyte here and the keratinocyte here. So this is the epidermis. And you can see in serial sections that melanin, especially in these 2 sections, melanin is secreted from the melanocyte and then internalized by the keratinocyte. And at some point, the melanin actually is not surrounded by any membrane. This is important because we see here a melanosome surrounded by the membrane, and then this membrane fuses with the plasma membrane of the melanocyte, and melanin is exocytosed and then internalized. So this and other, of course, other evidence that you can check in this publication led us to support this model of exophagocytosis as the predominant one in human skin. And this was only possible, as I said, by observing human skin biopsies. But of course, human skin biopsies are not amenable to manipulation. So we cannot silence genes or overexpress genes. So the workhorse of the field for many years has been the melanocyte keratinocyte co-cultures. We can study melanin transfer in these co-cultures because you see here a melanocyte and you see here keratinocytes around that internalize melanin. The melanin is produced and secreted by the melanocyte here, by any of these melanocytes, of course, in the culture, and then is internalized by the keratinocytes. And we use this model as well. And for example, we showed here you can see a melanocyte in green. There's a marker, it's a marker for that only stains melanocytes. And you can see a keratinocyte next to it that is not green. So that's how we distinguish a melanocyte from a keratinocyte. Of course, they have very different morphologies. That's another way. And you see here melanin got transferred along several days that we leave these co-cultures from the melanocyte to the keratinocyte. And we can measure the amount of melanin that was transferred, for example, here, by the area occupied by melanin, for example. And for example, using this system where we can silence proteins like this one called RAB11B, we saw that, for example, this protein is required for melanin transfer because if we silence it, melanin transfer is significantly reduced compared to the control. So these studies are only possible if you culture the cells and if you have cells that you can silence, you can transfect, etc., transduce. So it's not possible in human tissues. We using this 2D model, which was, as I said, the workhorse for many years. We found several of the proteins. We characterized this machinery. Several of these proteins are not showing the results because this is published, that are required for melanin secretion. And then we also find proteins required for melanin uptake using this 2D system. But of course, as it's obvious, and that's why we are here today, the skin is a complex tissue, is a three-dimensional tissue with a complex architecture, with layers, epidermis, dermis, etc. And so we really have to use 3D models. And that's what we started to implement in the lab a long time ago. This is the schematic, how we do them. We have two models. One is epidermis only, this one that I'm showing. We use the transwells or inserts with a membrane of polycarbonate. We coat with collagen, and then we seed primary human melanocytes. After that, primary human keratinocytes. We expose to the air-liquid interface, and with a special media with several factors that allow this differentiation into an epidermis structure, that after 2 to 3 weeks we have this structure formed. We also developed the model of the full skin equivalent in using polystyrene inserts. Here we see the fibroblasts first. The fibroblasts will secrete extracellular matrix, form a dermis, a layer like that is dermis-like. Then we do the same as in this one. We see the melanocytes. After that, primary human keratinocytes. These are primary human melanocytes. These are all cells, neonatal cells from humans. So, again, exposing to the air-liquid interface, we obtain this stratified tissue that in this case is composed by dermis and then epidermis layers, and in this case only by epidermis. Of course, dermis and epidermis takes much longer, obviously, takes around double the time. You can see here how this looks like in the histology. So the epidermis only, you see here the membrane cut in cross section, and you can appreciate all the layers of the epidermis: the basal layer, the spinous layer, the granular layer, and the corneal layer. All the layers are formed. So this is very similar to human skin. You can see here with all the layers that we can identify. And if we stain specifically for melanin, we find that melanin, for example, here a melanocyte, and the melanocyte is transferring melanin to the keratinocytes here, and the melanin forms the same supranuclear caps or similar to the human skin, like you see here. So it's very similar, has some differences. I will touch the differences towards the end, but in histology is actually remarkably similar. So here are some more stainings for tyrosinase. Tyrosinase is a melanocyte protein. It allows the synthesis of melanin, by the way. So you can see the melanocytes in the basal layer. Also S100 stains for the melanocytes. You can see here the melanocytes. The skin, regarding the skin model, the difference, as I said, is the dermis layer, composed by fibroblasts that secrete the extracellular matrix. And then we have a similar epidermis on top of the dermis with all the layers. The melanocytes are nicely aligned at the basal layer, and like in the epidermis, they transfer melanin, they produce and they transfer melanin to keratinocytes, and melanin accumulates on these supranuclear caps. So this, at least microscopically, with these stainings, is very similar to the human skin, as you can see on the right. Again, the stainings for tyrosinase to see the melanocytes and S100 melanocytes aligned very well here, the basal layer. So at least with these stainings, it's very similar to the human skin. What about the ultrastructure? So we did some electron microscopy and we saw both in the skin, RHPS means reconstructed human pigmented skin, or epidermis only here at the bottom. But we saw nicely this melanin, just like the human biopsies that I showed you, the melanin being transferred from a melanocyte. This is a dendrite in cross section to the keratinocyte, neighboring keratinocyte. And so melanin is transferred exactly in the same way as in the human skin. And these are reconstructed epidermis and the skins. And moreover, we saw that the melanin, after being transferred, melanin accumulates inside the keratinocytes in these granules that we call melanokerosomes, MKS, to distinguish from melanosomes, because melanosomes is the organelle inside the melanocyte, and these are different organelles already. And so you can see here, both in the skins or in the epidermises, these organelles that we characterized. They are surrounded by a single layer, single membrane, single bilayer. And this supports the model, again, of exophagocytosis. So this model nicely recapitulates the steps of melanin transfer that we saw in human skin biopsies and allows us to confirm this transfer model, and also validates the model, because we can find exactly the same route of transfer as we find in human skin. So we went further, as I said, and we looked at melanin inside the keratinocyte with these models. Now that we have them working, we can go a bit further, and here we see melanin fusing with lysosomes. Several examples of hydrolytically active lysosomes with this characteristic morphology, fusing with the melanin-containing compartment and forming the melanokerosome, that MKS that we saw previously. You see here several examples. So this was actually an interesting thing to find, because this was not known. It was not known if they fused or not with lysosomes. There was some speculation, but here clearly shows, these images clearly show that. And we also saw, interestingly, using this model, something that was never seen before, not even in the skin, because probably people didn't look thoroughly enough. We saw this tethering of the melanokerosomes to the nuclear envelope, here the nuclear envelope, and we saw these tethers, these membrane tethers, that we are now trying to characterize what exactly makes this membrane tethering. And so this, we think that we are trying to show that this is essential to keep the melanin close to the nucleus in the perinuclear area and form the supranuclear caps. So we are now trying to confirm that. But so this is the working model from this paper that we published last year. So melanin is phagocytosed, actually. This is already the keratinocyte, remember. Melanin is phagocytosed, the phagosome matures, the melanin-containing phagosome matures, fuses with lysosomes, forms this organelle called the melanokerosome, which is then transported to the perinuclear region and gets tethered to the nuclear envelope and keeps melanin in place. So this was only possible, these conclusions and this working model, by studying this reconstructed epidermis and skin models. So in terms of disease modeling, just a few slides to show you that we've been studying this Crucially syndrome for some time. This is a a hypopigmentary disorder. You see here characteristically some spots in the skin and the silvery hair or gray hair. Like you also saw, I showed it in the beginning, that small child with the gray hair. And this is known to be caused by mutations in any of these three proteins. So RAP27A, melanophilin, or myosin 5A. And what these three proteins do together, they form a complex, is they tether the melanosomes to the dendrites of the melanocytes, allowing the subsequent transfer to the keratinocytes. And so if you have a mutation in any of these proteins, what happens is that instead of having the melanosomes, this is the melanocyte, instead of having melanosomes spread throughout the cell, you have the melanosomes concentrated around the perinuclear area. When you image RAP27A deficient melanocytes, but the same happens if you do myosin 5A deficient or melanophilin deficient melanocytes. And this is, if you see the skin, if you look at the skin of a patient with this disease, you see this characteristic accumulation of melanin inside melanocytes. So melanin gets packed, concentrated inside melanocytes and cannot be transferred as efficiently, or almost no transfer, or at least you don't have an homogeneous transfer. That's why you have these spotty, these spots of hypopigmentation in the skin of the patients. We tried to model this disease in the lab, and for that we used the reconstructed epidermis, but instead of normal melanocytes, we used melanocytes silenced for RAP27A and normal keratinocytes, primary, all primary primary cells, primary human cells, and we could nicely recapitulate the accumulation of melanin inside melanocytes in this reconstructed epidermis. And this is very similar to what we see in the patients, the concentration of melanin, this big accumulation of melanin inside the melanocytes. Of course, if we have a model, we can study it in more depth. That's what we did. And here by EM, compared to a control epidermis, reconstructed epidermis, where you see again the melanin being transferred from a melanocyte to a keratinocyte, like we saw in the human skin. We have the Griscelli syndrome epidermis, where you see this big accumulation here, this huge accumulation inside keratinocytes of melanin, together with other vesicles, other material. And so, of course, this is not like the control. And this is probably one of the reasons why we see the phenotype in the patient's skin. And to cut a long story short, because this was a study done by João Chaneca, a PhD student. But to cut a long story short, what we found was an evidence for an alternative model of transfer. So remember, in the human skin, normal, let's say, we have this exophagocytosis. Now, when in this case of Griscelli syndrome, we think that this transfer route changes because the cell wants to get rid of the accumulated melanin, so it uses a different route as an emergency mechanism, basically to get the melanin, some of the melanin. It's never the same amount or same form of melanin, but still to get some to go across, to pass from melanocytes to keratinocytes. And that's why we saw this melanosome laden globules that we call being inside keratinocytes, like you saw in the previous one, instead of nice granules, as I showed you before, individual granules, usually, like I showed you before. Furthermore, we use this model to develop a melanoma skin, let's say, a cutaneous melanoma skin model. And this, instead of using melanocytes, normal melanocytes, let's say, we used melanoma cells, melanoma cell lines. Here on the left, you see an invasive melanoma cell line, A375, and on the right, non-invasive melanoma cell line. The invasive, you see that can invade, obviously, the dermis. You see here the melanoma cells in the dermis, and in this case, in the case of the non-invasive melanoma cells, you don't see the melanoma, the cells invading the dermis. Okay, and you can see the S100. You see melanoma cells, which stains melanoma, melanocytes and melanoma cells invading the dermis instead of which is the opposite of the WM115, which stay here in the basal layer and don't invade the dermis. And we use this to do some studies that are now published that you can see on the right, and we can measure differences by measuring the thickness of this invasion, let's say, by measuring the invasion through the thickness. So we are now developing, and this is the project we have in collaboration with MPS and funded by MPS, which is to do an immune competent skin, basically skin model, by adding immune cells to our reconstructed skin. We also have this perfusion model that mimics the flow, the blood flow, where we can use in which we can put the the immune cells, and the immune cells can circulate through the structure, through the inserts and the well where the skin is. And this will be used to ask this question. The project that we have is to test this hypothesis that melanosome exocytosis is enhanced in melanoma cells, and this promotes invasion and escape to the immune evasion, so escape to the immune system. And this is summarized here in this schematic. So our hypothesis is that melanocytes, or in this case melanoma cells, secrete melanin. That melanin is going to be to influence the tumor microenvironment, going to change it, and namely inhibit this immune response mediated by immune cells like T cells, macrophages, and monocytes. This will allow the melanoma cells to escape the immune response, and also allows the melanoma cell to become more invasive and basically in the end of the day metastatic. And so we are testing this using the 3D melanoma model that I was describing. So this leads me to the conclusions, and I just have one more slide of future perspectives or the current challenges. But basically we showed that this reconstructed human pigmented epidermis and skin models faithfully recapitulate the mechanisms of melanin transfer observed in human skin. They can be used to model pigmentary disorders like Shelley syndrome, we showed that, and also to model cutaneous melanoma and study the progression of this type of cancer and what is the role of melanin and melanin secretion in that progression. So they are a valid and excellent alternative to the use of animals in the research of skin pigmentation mechanisms and the defects that lead to pathology. And just to finish by adding these current challenges, of course, these models are still under development. Some of the challenges and some of the improvements the community is doing are, for example, that these models don't have what exists in the skin that is called the rat ridges. So those protrusions of the epidermis into the dermis, the models that I showed don't have that. But now with bioprinting, there are several studies that are mimicking these rat ridges. So what you see here, these protrusions with the bioprinting to allow skins with these rat ridges, because these rat ridges are important for the skin, basically rigidity and mechanical strength and resistant to mechanical stress. Also, these models usually lack skin appendages like hair or glands. And there's also an effort to add, for example, here you can see an example of hair follicles. So what was done here was to add hair follicle cells to the dermis. And so this is an improvement as well. And finally, the absence of vascularization. There are also several studies, just showing you one here of if you, this study added endothelial cells, and then the endothelial cells formed these, let's say, micro vessels. And it's something that is also being developed, and we would like also to improve our model and add some of these features, because of course it would be much closer to the skin, to the human skin, and to the human skin physiology. And so this leads me to the acknowledgement slide. It's important to stress that this model was co-developed with Abel Oliva from ITQB, also with the group of Miguel Seabra, and of course current and past members in the lab who were instrumental and funding, also from MPS NOVA, that allowed us to implement these models and currently use them and further develop them. And thank you very much.
Okay, thank you, Duarte, for the nice presentation. So questions?
My camera. Okay.
Questions? Anyone?
Yeah, I have a question. Maybe you mentioned it and I missed it because I'm a bit distracted today, I have to confess. But what do you think are the mechanisms of melanin changing the tumor microenvironment, or what is your hypothesis?
Yeah. So, yeah, thank you for the question. No, I didn't explain very thoroughly because of the time. But there are some studies suggesting that melanin outside the cells, the extracellular melanin in the tumor microenvironment, can be immunosuppressive. So suppresses the immune response. Also, if you think that melanosomes are big granules and are very rigid, melanin is a very tough moiety, let's say, and very rigid. The cells also have more trouble migrating, moving, and passing across narrow spaces to invade. So if they have melanin, physically they are more constrained and they cannot migrate. They are heavier, let's say, as well. So it impairs migration and invasion. So if they get rid of the melanin, they are in a better shape to migrate, to invade, and can also influence the immune response. So in terms of the tumor cell, of the melanoma cell, it's advantageous to get rid of the melanin, let's say like that. Mm-hmm. So now we are trying to study, of course, the mechanism and what happens, but also to modulate that and to stimulate secretion, to see if that enhances, and also to inhibit secretion, to see if that impairs the progression and invasion. So those are some questions we are trying to answer with these models.
Mm-hmm. And another question is, I think your model seems quite nice as you have it already. I mean, to study a lot of things. Do you plan to use it to study screen drugs, for example?
Yeah, definitely. That's an excellent question. We are trying to do that. Actually, we are doing that, but in a very small scale, very small throughput. So we are trying to increase the throughput first by increasing the number of samples we can do at the same time, the number of inserts we can do at the same time, and also to try to, let's say, to have readouts that are easy, fast, and at least medium throughput. So because right now we have to check by histology if the skin is good or not, and that's not high throughput, obviously, that's very low throughput.
Yeah.
So we are trying to do like more faster, simpler, more automatized, let's say, things that we can do to increase the throughput. But that's an excellent question. We would like to transform this in drug screening platforms. Thanks for the question.
Thank you.
Then actually I have a question
And
on the same topic. Have you tried in 96-well transwells develop this model?
Not yet. That's a very good question. That would be good. We have to get the inserts to the right size, but I think we could try, and we could try to adapt the number of cells and the protocol to a higher throughput format like 96 wells.
But because I heard from some other people working on skin models that the smaller dimensions, the media, or they are making this menisc disc more,
Yeah.
and this can cause some problems for the skin
Yeah.
models or not? Okay.
Yeah, actually, yeah. I think you are alluding to probably, so the pressure that the media makes outside the structure is very important, because if it makes a lot of pressure, it tends to the cells will get immersed, and the cells cannot be immersed. They have to be in the air liquid interface. So, and actually we tend to use bigger wells to decrease the pressure. You see that the media does. So it will be a challenge, no doubt. You are actually absolutely right. So we have to try different alternatives, but definitely this would be excellent if we can get a platform, a drug screening platform in 96 well plates, let's say. That would be fantastic.
Mm-hmm.
So it's worth the effort.
And then maybe last question from my side that I had received, and I actually don't know answer, maybe you have. Is there, in your opinion, difference, for example, if you take 6 well plate and turn the surface area into square and develop the skin model? Basically, the geometry of the field where you're growing this, in your opinion, will make a difference, or?
It could make a difference. I had never thought about that, to be honest. It could make a difference because even the size of the insert is important, and usually it's the let's say that the interface between the skin and or the epidermis and the well, the insert, where sometimes some of the liquid gets inside. You see, so that area, that frontier, that interface is actually critical. I'm not sure about the square shape, if it would help. That would be something, yeah, interesting. I don't know if it reduces the tension or increases. I'll have to think and maybe ask some physicists or so. But we could also try. We could also experiment. That would be probably even better. Thank you very much for the suggestion. We'll think about.
Yeah, okay, thank you.
Last question? No more? No? So thank you, Artus and Duarte, for your nice presentations, and thank you all for coming. And see you in our next webinar in February. And I wish to all happy Merry Christmas and Happy New Year. And thank you again for coming. Have a nice weekend. Bye bye. See you.
Thank you. Bye bye.